Hello, my lovely physiologists. Michele Glass here. Another chapter 12 video. What we're talking about first is what's described as graded potential. So the graded potential is sort of that, initial window of time when we are in some way stimulating the neuron, and then we kind of like do a wait and see, do we get to threshold or do we not get to threshold, right? And we've talked about this as like that kind of period of time when the team acetylcholine jumps in the chair, opens up the chemically gated sodium channel, and sodium team A initially comes in. And if we get the cell to threshold, then we boom, magically have our action potential. And so we've always talked about it in terms of depolarization, which is the stimulus. So this means that you are stimulating the cell. You're making it more positive. And so this is typically sodium or even calcium entering the cell. So in order to depolarize the cell, you make it more positive. And so we need a positive ion coming into the cell. This is what we mean by depolarization. And that's really the conversation we've been having, but that's not always the case of what's going on with our neurons. So we also see that we can do a hyperpolarization. And this would be more like an inhibition. So sometimes we need our nervous system to stimulate or signal tissue to do something, and sometimes we need to inhibit and tell that tissue to stop doing something. And so the hyperpolarization can come from either potassium leaving the cell, as we've talked about already, or it could also be from a chloride ion entering the cell. In the case of hyperpolarization, remember, you're making the cell more negative than resting. In the case of depolarization, you're making the cell more positive than resting, right? And our graded potential, remember going back, we said an action potential is all or none. The action potential got stimulated, got triggered when the cell reached threshold, which we said could either be negative 55 millivolts or negative 60 millivolts, depending on your reference materials. And the action potential, we said, was like flicking over that first domino. Once you get that ball rolling, there's no takebacks. With the graded potential, it's not in that way at all. A graded potential can be like really huge and dramatic and like real quickly take you to threshold. It can be a slow process. You may never get to threshold. And so we see a lot of interplay between this hyperpolarization and depolarization. That actually takes us to what we call the postsynaptic potential, or PSP for short. So postsynaptic is talking about the cell membrane. I'm going to say on the other side of the synapse. Cell membrane on the other side of the synapse. So you have the cell, and it's got the synaptic knob, and then you have on the other side of that the axon hillock or the dendrite. So the postsynaptic cell is the cell that's on the other side of the synapse. It's on the other side of the synaptic cleft. In the case of our chapter 10, it was the skeletal muscle fiber, right? So that's what we're talking about here with postsynaptic potential. What we saw too, let's draw our motor neuron real quickly. So let's do a circle for our cell membrane, and let's put our nucleus in there. Circle for our cell body and put our nucleus in there. And then let's do like our tree branch, like dendrites coming off. Let's do 3 or 4 of those. And then our long axon with axon terminals in the synaptic knob. What we see, remember, is you can have signals coming in from all these different dendrites, and they're processed together and integrated at that area called the axon hillock. And then that determines whether you get a signal sent or you don't. So when we're talking about the postsynaptic potential, we're talking about all these signals that are being received by the cell and what ultimately happens. Are you getting all depolarization signals? Are you getting all hyperpolarization signals? Are you getting a combination? What does that mean? So that's our question here. In the case of our postsynaptic potential, if you are getting depolarization signals, then we call this an excitatory postsynaptic potential. So this would be EPSP. EPSP. And if you're doing a hyperpolarization, we would consider this to be an inhibitory postsynaptic potential. So it's basically the same information we have on both sides of the screen, just sort of naming it in a different way. So the inhibitory postsynaptic potential can be abbreviated IPSP for inhibition. And the thing is that these two add together. They add together. So again, if you're getting a combination of excitatory and inhibitory signals, maybe it takes a longer period of time for you to get to threshold. If you're getting all excitatory signals, you get to threshold really fast. If you're getting all inhibitory signals, you don't get to threshold. If you're getting a couple of it excitatory but mostly inhibitory, you don't get to threshold. So it's this combination of how much excitement and how much inhibition you're receiving to determine whether that cell actually gets to threshold or not. So remember, the key thing is if you get to threshold, you get your action potential. When we talk about summation, we can have what's called spatial summation. And spatial summation is associating the activity of multiple inputs. So you're not getting the inputs at exactly the same time, but you're getting those inputs and you're putting them together to determine whether you get to threshold or not. In the case of our temporal summation, you're getting multiple action potentials from a single cell. All right, that's it for this video. Stay tuned for discussion on synapses. And as always, take care of yourselves and each other.